Hi, I'm Madison Zapata, and this is my scientific writing protein presentation. Hemoglobin is my protein. So, hemoglobin serves to transport oxygen into red blood cells. It's present in all forms of life, eukaryotes, prokaryotes, and archaea. It's made up of multiple globular subunits that make up its quaternary structure, with alpha helices and heme groups in each subunit. Defects in red blood cell shape may have a negative effects on the body by causing mutations, such as sickle cell anemia and thalassemia. Hemoglobin is located in all red blood cells. There are roughly 270 million hemoglobins present in all red blood cells to carry out oxygen throughout the body. It carries oxygen O2 starting from the lungs to tissue and all of the organs, and then carries carbon dioxide back to the lungs, where it can then be exchanged for oxygen. The 3D ribbon model shown here is the protein composed of several subunits. There are a total of 4 subunits, 2 alpha globins and 2 beta globins. As seen in the figure, you can see that there are 2 betas and 2 alphas. The secondary structure is a regular and repeated folding of the polypeptide chains that make up alpha helices. Here you can see the alpha helices, with each subunit containing around 8 alpha helices. All these alpha helices are bonded together in 4 subunits to form the tertiary structure, which is the entire 3D formation formed by polypeptide chains. These 4 subunits are all bonded together to make up the quaternary structure of the protein. Also, in this figure, you can see the heme groups, which are these green little units here. Through gene sequencing conducted on the protein done by researchers, they have concluded that the gene of hemoglobin consists of 10 genes that are in 2 groups. The 4 main subunits make up the protein are alpha globins and beta globins. The alpha globin gene sequence is tightly at the chromosome position 16p 13.3 in order HBZ, HBM, HBA2, HBA1, and HBQ1. The beta globin gene sequence is at 11p 15.5 in the order of HBE1, HBG2, HBG1, HBD, and HBB. The heme group, which is this figure here, has an iron in the middle, shown right there in the middle. The iron cations with these 2 bonds bind to the nitrogen with covalent bonds. These are represented by the solid black line. So it's bonded here with these solid black lines. Non-covalent bonds are with the other 2 nitrogens represented with the red dotted lines. Those are here. The iron molecule is essential in allowing the protein to keep its shape. The heme provides the protein with a binding site for oxygen. The binding between the iron molecule and the oxygen molecule occurs because iron has a positive charge and oxygen has a negative charge. These opposing charges allow the binding to occur. However, this bond is unstable and it is a reversible bond. When bonded to oxygen, it is oxyhemoglobin. That oxygenated state is a bright red color. When iron is not bonded to oxygen, it is deoxyhemoglobin. It's in its reduced state with a dark, deep red color. The heme has hydrophobic and hydrophilic regions. The 2 areas interact with water on the exterior of the globin, but the rest of the heme group does not like water, so it's hydrophobic and therefore stays on the inside of the globin. Additionally, since the bond with iron and oxygen is reversible, the protein unbinds with oxygen when the body needs the oxygen. For example, while running, carbon dioxide will bring hemoglobin to allosterically inhibit oxygen from binding in an acidic environment. Within the red blood cells, it contains carbonic anhydrase that disassociates the carbon dioxide to carbonic acid. This action releases protons, which are then able to allosterically inhibit oxygen, from binding and changing its shape to the hemoglobin and releasing oxygen where it's needed in the body. Hemoglobin is also the protein that gives red blood cells its pigment. This pigment can be seen in many other areas. When the red blood cell dies, hemoglobin is broken up and then it's released. The heme is transferred to bone marrow and the rest becomes bilirubin. Bilirubin is a chemical that's excreted from the liver to the bile and then the intestines, where it can exit the body and give feces its brownish color. Sickle cell anemia is a genetic disease associated with hemoglobin, where red blood cells do not assume its normal shape. The red blood cell is sickle-shaped and causes cells to become hard and sticky. This shortens the red blood cell survival time and produces anemia. Decreasing in red blood cells. Anemia can have many effects on the body, such as delayed growth and development, shortness of breath, exhaustion, and in some cases, jaundice. Sickle cell anemia also causes blood clots that do not allow for the passage of blood through small blood vessels. By blocking passage of blood, it does not allow for oxygen to travel throughout the body, and this can produce damage to many vital organs as well as pulmonary hypertension. These blockages can also have people experience pain. Thalassemia is another genetic disease that's associated with hemoglobin. It's an inherited disease that has many different forms. This disease results in a decrease of red blood cells and hemoglobin, which then also produces anemia. Due to the decrease of hemoglobin, iron has limited areas to bind. If there's an excess amount of iron present, it can cause damage to many major organs, such as the heart and liver. Thalassemia can be minor and may not need medical attention. However, it can be more severe, needing regular blood transfusions conducted to keep levels normal. When thalassemia is more severe, it can affect the body's bones from not forming correctly, and enlarged spleen, slow growth rates, and then again, heart issues. In conclusion, hemoglobin is a complex protein that serves a vital role. It's essential for human survival and is seen in all other forms of life. Mutations in this protein cause life-altering effects like sickle cell anemia and thalassemia are just a few of these altering diseases seen in the mutation of hemoglobin. Thank you.